Good afternoon, everyone. We'll be starting the webinar in a few moments. Good afternoon. Our webinar this afternoon is the start of a webinar series that the Natural Sciences Research Institute holds annually. This year, NSRI's lecture series consists of quarterly webinars, which are open to the public and free of charge. We're very happy to note that there are numerous schools and agencies that have registered to join us this afternoon. And to name a few, there's UP Manila, UP Diliman, even some as far as Western Visayas State University, Angeles University, University of San Agustin. We have several from Philippine Science High School, Muntinlupa National High School, the University of Rizal System, from the agencies of DepEd and PAMET, universities like PUP, CLSU, St. Louis University, PLM, and so many more. We're so happy that you would join us this afternoon. Just a few minutes more before we start, I'll just read a few reminders. We ask everyone to keep your microphones muted and video cameras off during the webinar, unless you have been asked to turn them on. There will be a photo documentation session at the end of the webinar. Pictures from this photo session may be posted on the NSRI Facebook page. For data privacy reasons, we ask those who do not consent to be photographed to keep their cameras off and to remove their profiles pictures. Of course, if you'd like to join in the photo documentation, please turn your video cameras on when the photo session starts. Also, questions will be answered during the open forum after the second talk. Please type your questions in the Zoom chat box. And for those who are viewing the webinar via YouTube, use the Google link located in the YouTube description box to type in your questions. The link to the evaluation form will be given later during the webinar. You'll have to fill out this form within the day to receive your e-certificate of attendance. Of course, we'd all like to have our e-certificate of attendance, so make sure that you click on the link and fill up the evaluation form. Your e-certificate will be sent to the email address that you provide. And now let's begin the webinar. To open this afternoon's webinar, we are fortunate to have with us a physicist whose field of specialization is in terahertz optoelectronics, semiconductors, and optical spectroscopy. His research interests include terahertz time domain spectroscopy, ultra-fast optical processes, and semiconductor optics. For me, that sounds very highfalutin. His work was recognized by the National Research Council of the Philippines, or the NRCP, with the 2018 NRCP Achievement Award in Physics. He obtained his PhD in physics from the National Institute of Physics, or more often referred to as the NIP. This institute is part of the College of Science of the University of the Philippines, Diliman. And now he is currently a professor and researcher with the NIP. He was previously program director of the Material Science Engineering Program, or MSEP, and is deputy director for facilities and resources of the NIP. Quite significantly for us here at NSRI, he is now also the director of our institute. To give the welcome remarks, Let us give a hand to Dr. Elmer S. Estacio. Sir Tux, the floor is yours.
Thank you. Thank you very much. All right. So, good afternoon, everyone. I was requested to give this welcome address today, and I accepted the invitation as this would be a good chance for me to introduce myself as the NSRI director. Ako po si Elmer Estacio, and I'm a member of the faculty of the National Institute of Physics, UP Diliman. This webinars, as I've heard, is the second installment of a series of webinars that kicked off last October and will hopefully be conducted quarterly or is planned to be conducted quarterly. I have heard that the first one was a huge success, and this webinar that we will be having today is also well subscribed, as was described earlier, with registrants coming from a very broad spectrum of participants, from high school students to experts in their chosen fields, and coming from all over the Philippines. I applaud the NSRI staff under the leadership of Dr. Jonas Kilang for their hard work in organizing these webinar series under these pressing circumstances and under the mandate of the NSRI in conducting symposia and seminars for the dissemination of new knowledge in the natural sciences. So, with that, I'm not going to keep this long. I welcome our two guest lecturers for this afternoon, Dr. Hil Peñular and Dr. Marie Christine Obusan from the Institute of Biology, UP Diliman. And of course, I welcome all of you to the NSRI Lecture Series of 2021. Good afternoon. Maraming salamat po. Thank you.
Thank you, sir, so much, Sir Tux. So, the NSRI seminar series or lecture series or webinar series is this year celebrating some past researches that have been conducted in the Institute. In 2017, the Department of Science and Technology Philippine Council for Industry, Energy, and Emerging Technology Research and Development, or DOSTP shared, granted the Natural Sciences Research Institute funding to support researches that would progress strategic areas that are significant for the country's development. Our two speakers this afternoon were recipients of research funding under this program. Our first speaker this afternoon is Dr. Marie Christine M. Obusan. She is an assistant professor of the Institute of Biology of the College of Science in UP Diliman. She obtained her PhD in environmental science from the Institute of Environmental Science and Meteorology, or IESM, also the College of Science in UP Diliman. She is principal investigator of the aquatic lab of the IB's Microbial Ecology of Terrestrial and Aquatic Systems, or METAS laboratory. Her work is on the microbial ecology of aquatic systems with focus on sentinel species and habitats. Her current projects include the use of microbial communities as indicators of emerging environmental pollutants in water resources, biotechnological potential of bacteria from stressed environments, and emerging diseases of cetaceans. In 2017, she was granted a shared NSRI research grant to study polyethylene degrading and lactase producing bacteria from highly polluted MMORS, future application for industrial and environmental in situ pollutant degradation. That's quite a mouthful. Here to discuss with us the degradation potential and enzymatic activity of bacteria from the highly polluted MMORS, which by the way means Marilao, Meycauayan, Obando River System. Let us all welcome Dr. Marie Christine M. Obusan. Dr. Obusan, you have the screen.
Good afternoon.
Afternoon.
I will share my slides. Okay. Thank you for the introduction, Ma'am Miriam. Good afternoon again, everyone. I would like to thank NSRI for this opportunity for me to share our work on the degradation potential and enzymatic activity of bacteria from the highly polluted Momors. Now, as mentioned by Ma'am Miriam, Momors is Marilao, Mekawayan, Obando River System. Medyo mahaba. So, it's the name of a river system, and some of you may be already familiar with this river system because it is notoriously known for being highly polluted. Before I discuss the details of the study, I would like first to acknowledge the following institutions which help us in our project implementation. First, the DOSTP shared in collaboration with NSRI for the project grant. And then, of course, the Institute of Biology, or IB, my lab, the METAS lab, the Philippine Genome Center, and the Region Lab and the the Plant Genetics and Cyanobacterial Lab, both labs in the Institute of Biology also which help us in our project implementation. And I would like to acknowledge also Mr. Arwin B. Verdera. He is my advisee and a part of my research team in METAS lab. METAS is Microbial Ecology of Terrestrial and Aquatic Systems Laboratory. Arwin worked with me in this project, and the data that I'm going to present today are actually generated from the experiments that he conducted. This is also a part of his MS thesis, and I'm happy to share that Arwin finally defended his master's thesis last December 18. Okay, let me start by introducing the project site. Momors is a 130-kilometer long waterway that empties into Manila Bay. Upstream, we can find industries and agricultural areas, and then downstream, we can find small and commercial fish ponds. Of course, there are also residential areas. Momors is included in the top 30 dirtiest river in the world as reported by the Blacksmith Institute, and several studies have confirmed that it is heavily polluted with plastics and heavy metals, probably from the effluents coming from industries. Some of the industries that were identified are lead acid battery recycling, gold and precious metals refineries, and then we have also tanneries. Despite the pollution, Amomors is classified as Class C river water, which means the water can be used for fishery, for recreation, such as for boating and fishing, and also for agriculture, such as for aquaculture, and for irrigation and livestock watering. Momors actually supports Bulacan's aquaculture industry with the production of the species that you can see on the screen. And this is a point of concern because many studies also reported that the water parameters of Momors exceed the safe limits and standards of the Bureau of Fisheries and Aquatic Resources. So since Momors is a highly polluted body of water, it is considered a stressed environment. Because of the presence of pollutants, you would expect a different set of physicochemical parameters or conditions of the water and also the sediment, which serve as sinks for those pollutants. Now, the pollutants in turn serve as substrates for microbial growth. And it has been proven in many studies that substrate availability is highly associated with the presence of genes in microorganisms that code for enzymes that have the ability to degrade the substrates available. And that is part of microbial tolerance and adaptation mechanisms. So in the case of Momors, we can expect that the bacteria and other microorganisms thriving there may produce metabolites that can be explored for different applications. And in our case, biotechnological applications. So this is the framework from which we have based the project objectives. So in general, we aim to screen bacteria for polyethylene or plastic utilization. to evaluate the degradation potential and associated enzymatic activity of our candidate bacterium or our best isolate, and to characterize our candidate bacterium. Now, let me show you our sampling stations. We specifically collected sediments from the Mekawayan River, tributary of Momors. So we have 5 sampling stations starting from the upstream. You can see here photos of our sampling stations. So we have first the Viente Reales, which is near plastic industries. Next, we have the Kainin station, which is near tanneries. We actually saw the tanneries there. And then next, we have the MacArthur Bridge. You can see there the waste and the debris, which is downstream of Kainin. And next, public market, where we found gold smelting industries. And the last sampling station is the upstream of the mixing zone, which is the downstream of the public. We conducted our methodology in 3 phases. So I will explain briefly the phases. In phase 1, we directly screened the collected sediments for the presence of bacteria that are able to utilize polyethylene. So to do this, we use polyethylene as the sole carbon source of bacteria in the medium, and then the bacteria that grew, survive, that grew and survived were isolated, purified, and their genotypic identifications were confirmed through 16 sRNA gene sequencing followed by phylogenetic analysis. Now, from among the bacteria that we have screened, we selected the best isolate, or we call now the candidate bacterium, and then we tested that isolate for further biodegradation activities. So first, we conducted the so-called pH degradation assay, wherein the activity of the isolate against low-density polyethylene and high-density polyethylene was tested. And this also involved scanning electron microscopy imaging. This assay was followed by another assay, the Guayacol assay, which helped us confirm if the polyethylene degradation activity is caused by lactase, which is a type of enzyme. And then our candidate bacterium was also subjected to Folin-Schucalto assay to test its ability to degrade BPA. This was followed by ABTS assay for us to confirm if the BPA degradation activity, if exhibited, is caused by phenol oxidase, which is another type of enzyme. Now, we observed that our enzyme is producing a brown-black pigment. And so we added this phase. So in this particular phase, we performed pigment production assay followed by pigment extraction and purification. After that, the extracted pigment was subjected to chemical characterization such as solubility tests, precipitation tests, a determination of absorbance, and so on. And we also perform DPPH assay to see if our pigment has antioxidant activity. Now, concurrent to pigment extraction, we also perform tyrosinase activity assay. Now, the goal of this assay is to detect dobachrome formation. The presence of dobachrome will help us confirm if the pigment that we have is a melanin pigment. So, we hypothesize that it is a melanin because of its color. The presence of dobachrome will confirm the activity of another type of enzyme, which is tyrosinase. So very briefly, tyrosinase catalyzes the production of melanin, wherein dobachrome is one of the intermediates. In phase 3, we characterize our candidate bacterium through whole genome analysis. So we submitted our sample to the Philippine Genome Center, wherein they performed the sequencing, and then we obtained the raw reads. We filtered the reads to remove the low quality reads and bases, and then we performed de novo genome assembly. And then we assess the assembly in order to determine if it is good enough for downstream analysis. That was followed by taxonomic classification, wherein the closest taxonomic relative of our isolate was determined using the MIGA online database. So we use several parameters such as, for example, average nucleotide identity, average amino acid identity, and other parameters. And then the last step is the annotation, wherein in simple terms, we identified the genes present in the genome, and also we perform an analysis using a separate software to identify the so-called BGCs, or biosynthetic gene clusters. These are the genes coding for secondary metabolites. Now, I will be presenting the result highlights. I'm sorry if I will not discuss some details of our results and methodologies in consideration of publication concerns and possibility of patent applications. So to start with the result highlights, shown here are the physicochemical parameters of the water among the different sampling stations. So in general, no, I determined that there were no significant differences in the pH, temperature, dissolved oxygen among the sampling stations. But we observed an increase in salinity, total dissolved solids, and conductivity as we go from upstream to downstream. In relation to this, it is important to note that we isolated more bacteria from the upstream region where these parameters are lower. A similar study reported that the accumulation of heavy metal in the area is from upstream to downstream, and heavy metal may be a possible factor that affects microbial diversity and abundance. Also, considering chemistry, conductivity, and the presence of ions may also affect the mobility of heavy metals. Here are the bacteria that we have isolated. So these are the polyethylene utilizing bacteria from the sediments. We have isolated a total of 26 bacteria, and you can see here the codes and the genera. Overall, these bacteria belong to 8 different genera. So among the collection of isolates, we identified isolate H28 as our candidate bacterium for degradation and enzymatic activity evaluation. So we consider this as our best isolate because it is able to utilize both low density and high density polyethylene. As for the result of the degradation assay using polyethylene films, you can see here the data on the percentage weight loss. So in general, there are differences in the percentage loss among the negative control and the different days of incubation of the plastic films with our isolate. So we have 60 days, 90 days, and 120 days of incubation. But when we tested our data, we did not confirm a statistical significance. However, you can see here from the presented electron micrographs that there are surface abrasions. So N is our negative control, and you have here the numbers representing the days of incubation. So you can see here that some parts are eroding, and there are also some holes specifically in films incubated for 60 days and 120 days. So the same is true in the case of degradation assay using low density polyethylene. So even though there are differences, when we tested the statistical significance, wala kaming nakuha na significant results. So in general, there were no significant differences among the percentage weight loss in negative control and in the different days of incubation. However, the scanning electron micrographs show not only surface abrasions but cellular structures which we're still investigating in relation to bacterial colony formation. And I have mentioned that isolate H28 is also producing dark pigment. And in general, we found out that the pigment production is different. The pigment production differs depending on the type of media that we use and also depending on the supplements that we introduce into the media. The absorbance spectrum also help us confirm the type of the pigment that we have. So based on the UV visible absorbance spectrum showing peak at 230 to 260 nanometers, we confirm that our pigment is indeed melanin. And in addition, the Fourier transform infrared spectroscopy confirmed that it is a particular type of melanin, which is eumelanin. So for the chemical characterization, we have results which are consistent with what's reported in the literature. For example, in the case of solubility with the different treatments, we found out that the pigment is insoluble to acetone, to methanol, and so on and so forth. And we also found out that it can be precipitated in the presence of copper, iron, magnesium, and manganese, which is also consistent with the results of other studies. So in general, we confirm that isolate H28 produces melanin, and the production of melanin in the process of melanogenesis is catalyzed by tyrosinase enzyme. Now, we have our established set of parameters for the bioproduction model, and we're still optimizing until now in order to increase the yield and enhance the tyrosinase activity. You can see here the melanin pigment produced. And also, the pigment that we have has antioxidant activity based on the DPPH assay. So this is in comparison with the activity of the positive control, the gallic acid, and the negative control, which is the DMSO. As for the whole genome sequencing, the project was already deposited at GenBank. If you want to access the document with all the details, you can use these accession numbers, and you can also use the bioproject number, biosample number, and the sequencing read number shown in the screen. Sorry. Okay. And in general, also, we found out that our isolate belongs to a species that is not yet represented in the Microbial Genomes Atlas online database. And thus, it is a possible novel species. So I'm happy to share also that we have produced a publication from this project. The draft genome sequence of isolate H28 is already published in the Microbiology Resource Announcements of the American Society for Microbiology. So in this paper, we also reported that there are different genes coding for proteins which are associated with plastic degradation. So these are the esterase, lipase, multi-copper oxidases, and hydroxylases. There are also genes coding for putative tyrosinase and tyrosinase cofactor, which is associated with melanin production, as I have explained. We also detected 30 biosynthetic gene clusters, or BG, which are coding for 13 secondary metabolites, including melanin. So in summary, from the Mekawayan River sediments, we isolated 26 polyethylene utilizing bacteria belonging to 8 different genera, which you can see here on the screen. And this bacteria did not show any BPA degradation activity and oxidase and lactase activities. Our best isolate H28, is a possible novel species, and it degrades both high density polyethylene and low density polyethylene. It also produces melanin with antioxidant activity and possesses biosynthetic gene clusters that code for 13 secondary metabolites. For further studies, we hope to observe surface changes earlier during the incubation period in the polyethylene degradation assay. We're also planning to conduct further studies with longer incubation periods, and that we aim to elucidate the mechanism of polyethylene degradation. And for other researchers, it would be good also to try a consortium of bacteria for more efficient degradation. We also plan to explore our melanin for its uses. Melanins in general are used in phenol degradation, in reactive oxygen species scavenging, in heavy metal chelation, and also they are being tested for their cytotoxic and anti-proliferative activity in relation to the development of drugs for cancer. So with this, I would like to end my presentation. Thank you very much for your kind attention. If you want to email me your specific questions, or if you're interested to get a copy of our publication, you can contact me through my email address, and you have there the Facebook page of our laboratory and our website. Salamat po.
Thank you so much, Dr. Obusan. Can we have a round of applause through the reactions button? Here we have a reactions button. Yes. Thank you so much, Dr. Obusan. That was quite interesting. Hindi pala MMORS, kundi MOMORS dapat. And who knew that so much potential was there in highly polluted areas? That's quite interesting. Thank you for sharing with us.
Salamat po.
Everyone, we will be having a 5-minute break. But before that, I would like to remind everyone that questions will be answered during the open forum after the second talk. So type your questions in the Zoom chat box or use the Google form link located in the YouTube description box to type in your questions for the open forum. Yes, questions will be sorted and collated so that questions of the same kind can be asked at the same time. Now, let's take that 5-minute break. See you in a while. Welcome back, everyone. Welcome back. I hope everyone had a good break. Another interesting talk awaits us. But before we begin, I'd like to recognize some of the other participants of this webinar who I haven't mentioned yet. For example, those from UP Los Baños, University of Immaculate Conception, Quezon City Science High School, CEU, RITM, and even the Department of Health. I'm sorry if I left anyone out, but to all of you, we're very glad that you were able to join us this afternoon. Moving on, our second speaker is Dr. Gil M. Peñular. He is an associate professor with the Institute of Biology of the College of Science here at UP Diliman. He obtained his PhD in medical science from the Gunnam University Graduate School of Medicine in Japan. He is principal investigator and supervisor of the IB's Medical Microbiology Laboratory, which is a biosafety level 2 facility where basic and applied researches on probiotics and on microorganisms of industrial and medical importance are conducted. His 2017 shared NSRI research was on the microbial prospecting of Mount Banahaw for actinomycetes with pharmaceutical industry applications. To speak to us this afternoon about the cellulase and antimicrobial activities of Bacillus species and Streptomyces species, which were isolated from an urban green space in the Philippines. Let us all welcome our second speaker, Dr. Gil M. Peñular. Dr. Peñular, the screen is yours.
Thank
Good
you.
afternoon.
Afternoon. Okay.
Wala pa, sir.
I cannot share my screen.
Maybe we can share from here. Sir Jude.
Na-stop share ko na po.
Can you try sharing the slides of Sir Gil?
Wait. Co-host na naman ako, di ba?
Yes, sir. Opo.
Right. But for some reason, when I click basic, the only thing I see is the whiteboard. Whiteboard. Wait lang.
Okay. Sir, baka gusto niyong isend yung slides niyo?
Yeah, sure.
Opo. Para we can play it from here.
Okay. Kanino ko isa-send yung presentation? Sorry for the delay, guys.
Okay lang, sir. Kay Ate Vina po.
Hmm.
Pwede rin
Or,
akin if you want. I can manipulate the slides for you.
thank you for offering, ma'am. Well, everyone, while we're waiting for Dr. Hill, these technical problems happen. We all know that. Think about the questions you'd like to direct to Dr. Obusa and use the chat box dito sa Zoom or the link to the Google form if you're using YouTube, watching from YouTube. Marami tayong natutunan kanina dun sa talk ni Dr. Obusan. And actually, there are already some questions that have come in, interesting questions. I'm sure Dr. Ubusan will be able to address these questions later on. I may as well take this opportunity to remind everyone to keep your cameras off and your microphones off during the presentation. Here we go.
Can you see my screen? No.
Yes, sir.
Okay, okay na. Sige. Thank
Yes,
you.
sir. Opo. Thank you, sir.
Okay.
It's Gmail.
No, dito sa screen wala pa rin.
Dito po sa screen, Gmail po yung nakikita namin.
Gmail. Wait lang.
Well, I think it's Gmail.
Wait. How about now?
Ngayon po, wala pong screen
Wala.
sharing.
Now, meron na.
Yes, it's the Quezon Hall.
Okay, so kita na yung slides.
Yes, sir.
Okay, so I can start.
Hindi pa po yeah, there you go.
Okay.
Thank you.
So sorry, terribly sorry for the technical delays, but thank you for the introduction and good afternoon, everyone. I'd like to acknowledge the NSRI Seminar Committee for being patient with me because of the technical difficulties, and of course for inviting me to share the results of my NSRI Fishery project. Now, this work was conceived and executed in collaboration with one of my graduate students, Mr. John L.D. Dakiwag, who did all of the experiments as part of his master's thesis. So I think Joma is part of the participants right now. So good afternoon to you, Joma. So let's start. So it's almost been a year, okay, since Luzon or Metro Manila has been placed under community quarantine as a response to the COVID-19 pandemic. Okay, so of course, quite recently, the IATF has been modifying their guidelines. Now, these modifications might be an indication that things are somewhat improving and that key issues with regards to the pandemic are being addressed. We have to remember, however, that before the COVID-19 pandemic, there were already a lot of global crises and issues that unfortunately were somewhat put aside because of the pandemic. So you don't see a lot of news about climate change, about global warming and loss of extinction right now in the news and in the social media. Now, some of these global issues are quite overt, meaning you can actually feel their impact almost immediately, or it impacts a lot of individuals and a lot of countries, like, let's say, fuel shortage, while some global crises are more insidious, meaning yung development nila are somewhat gradual and subtle, but still, no, yung effect nila would be quite harmful, and the consequences are quite severe as well. Okay, so okay. Now, of course, going back to a fuel shortage, a lot this basically this is when the supply doesn't meet the demands. Okay, and this could be due to a lot of reasons. Let's say the global market, infrastructure problems, if there are pipes that are damaged, or management issues, if there are strikes in fuel factories, or ultimately this could be due to the very nature of the fuel itself, which is non-renewable. Now, in the case of antimicrobial resistance, this is when microorganisms change over time. And because of certain mutation or changes, they no longer respond to conventional treatment. Now, this would be a serious problem in the medical field because it would make the treatment of infectious diseases more difficult and would definitely increase the risks of, let's say, transmission, disease, and even death. Now, as an indication that the problem of antimicrobial resistance have become more severe, is now we have a lot of terms that go beyond multi-drug resistant microorganisms. We have extremely drug resistant organisms, or XDR, and even pan drug resistant microorganisms, or PDR microorganisms, that are practically resistant to all of the antibiotics we have at the moment. Okay, but similar to us, okay, of course, we have when we have problems, we have our go-to individuals, our parents, our friends, and our colleagues. So similarly, for these global issues, one of the best sources of solution would be nature, particularly the microorganisms present in nature. So, for example, in the case of fuel shortage, uh, recent developments have came up with a biofuel like bioethanol and biodiesel, which are derived from renewable resources but are converted to these biofuels through the use of microorganisms that act as biocatalysts or bioprocessors. Shown in this table would be a short list of microorganisms currently in use, okay, to produce ethanol and biodiesel from various substrates. Now, this particular paper published in 2019 shows the use of bacillus in the production of biofuel from sugarcane molasses. Okay. Okay. Now, going back to this figure, so you can see from the third column that you can actually have a lot of substrate for bioconversion to biofuel. So from simple sugars to more complex materials like cellulose. And cellulose in particular is quite significant because this is the most abundant organic compound on Earth. And you have different sources of cellulose, from plant material to agricultural waste. Of course, you also have industrial waste made of cellulose, and of course, food waste as well, like mga apple peelings. A lot of these also contain a lot of cellulose. Now, what I want to say at this point is that these materials, especially the waste materials, can be used as a substrate for bioconversion. So you can use the cellulose from these materials, uh, enter them into a process that would eventually convert them to ethanol. Now, for an organism to do this, or for an organism to be useful in the bioconversion of cellulose to biofuel, it must have the following enzyme. It must have cellulase, which consists of at least three enzymes. Cellulase is a multi-enzyme system consisting of several enzymes that sequentially attack a biomass, particularly cellulose, slowly converting it into more simple material, but eventually to glucose, which can be fermented to biofuel. Okay. Now, indicated on this slide would be two species of bacillus that have been studied to contain cellulose that are potentially useful in industry. Now, with regards to antimicrobial resistance, again, the main problem with this is that it makes the treatment of microbial pathogens more difficult. And according to one projection by 2015, about 10 million deaths would be due to diseases that cannot be cured because of antimicrobial resistance. Now, most of the antibiotics we have right now are derived from microorganisms. Of course, there are other antibiotics that are semi-synthetic, which is basically still derived from natural products, or completely synthesized antimicrobial, but still their structures are more or less patterned after those derived from nature. But overall, most of the antibiotics we have right now, more than half, is still derived from microorganisms. And among this, about 67% is actually obtained from one single genus, and that would be Streptomyces. Now, because of the limitations of synthetic of antibiotics, the main problem kasi with synthetic antibiotics is that although they are very effective in vitro, sometimes their application in real life is not that simple because there are usually problems with how these synthetic antimicrobials enter the cell. So there is a value in searching for natural products that could address the current problems of antimicrobial resistance. And similar to this study presented here that was published in Nature last year, they were able to identify a novel antimicrobial called carbomycin from this particular species of Streptomyces. Now, this particular antimicrobial attacks the cell wall, but unlike penicillin, which basically prevents the buildup of cell wall material, carbomycin prevents the binding of autolysine, which means that the cell wall cannot remodel itself. And of course, for students of microbial physiology, you should know that for a cell to grow, there should be an interplay of cell breakup and buildup. So since the cells cannot divide, okay, it eventually leads to cell death. Here are some additional compounds recently discovered or at least being studied as new antibiotics to combat antimicrobial resistance. Now, of course, when it comes to the environment, there are different types of environments, and one of the things that is currently being explored would be the green spaces. Green spaces basically refer to any piece of land that is partially or completely covered by vegetation. So even in urban areas, we have green spaces. And indicated here are several examples from community woodlands to parks and cemeteries. Now, these urban green spaces are classified into different types based on these categories, based on the amount of plant cover, based on aesthetics and amenities. Now, the University of the Philippines at Diliman is one of the largest green spaces in Metro Manila. And within the university, you can identify distinct locations where green spaces somewhat differ in their characteristics, but not too different. Okay. So, Mr. Dakiwag and I posed this question: can we actually isolate bacteria, okay, with cellulase and antimicrobial activity from these green spaces that can help address the issues that I have mentioned before? So this brings us to my presentation for today. That would be cellulase and antimicrobial activities of Bacillus and Streptomyces isolated from an urban green space in the Philippines. So we have four objectives for this study. That would be to isolate bacteria from green spaces and then screen them for anticellulase activity, screen them for antimicrobial activity, and for isolates with promising activities, characterize them and identify them. Okay, so for the first objective, we had to collect samples, or we decided to collect samples from four sampling sites within UP Diliman. Okay, and these are the coordinates of our sampling sites. And before we can proceed with the sample, we had to ask permission from the office, from the office of the Vice Chancellor of Community Affairs. Now, once we got that permission, we proceeded with the sample collection. Okay, so After sample collection, we air dry the sample and then sieve them. And then afterwards, we proceeded with the treatment. So aside from untreated soil sample, we treated parts of the collected sample with 1.5% phenol and then exposed the other samples to wet heat. So practically, it's about 65 degrees Celsius in a water bath. Okay, and then afterwards, we performed a serial dilution. We mixed the soil with sterile saline and then diluted that up to 10 to the negative 5 and then plated that to three different culture media. Now, initially, the goal of this study was to isolate actinomycetes. No, but based on our readings, aside from actinomycetes, we can easily also isolate bacillus and streptomyces from the sampling sites that we have decided. Now, these three culture media have been supplemented with nystatin, which will serve as our antifungal agent. Okay. Now, after incubation, we obtained colonies. So we chose colonies that were dry, with and without pigments, and with aerial and substrate mycelium. So basically, colonies that looked like actinomycetes, bacillus, and streptomyces. Now, for the results for this particular objective, so we were able to collect 25 soil samples. 25 soil samples because for each sampling site, we decided to take soil samples from 5 different locations. So the goal here is to get a better representation of each sampling site and hopefully isolate more microorganisms. But after processing, after drying these soil samples, for practicality, we had to pull them together, at least those that belonging to the same sampling site, before we proceeded with a serial dilution. Now, these are just representative plates of our spread plate. Notice that the colonies are of different sizes, different colors, different textures, which is understandable because the soil is a rich environment for different microorganisms. And these are representative plates of our purification. Notice the cultural characteristics of the pure colonies, which would be indicative of either actinomycetes, bacillus, or streptomyces. Now, for the results, we were able to obtain 385 distinct isolates from all of our sampling sites. Now, this is true. This is understandable because again, the soil is a rich microenvironment where microorganisms are found in abundance. There was an estimate that about 10 billion microorganisms per gram of soil, meaning you can isolate, you have that amount of microorganisms per gram of soil. And yet we only found 385. Now, this is probably due to what is called the Great Plate Count anomaly, which simply means that our culture conditions cannot provide all of the things necessary to grow all of these microorganisms. So whenever you isolate microorganisms from the environment, there are limitations. You are limited by your culture media, by your physical factors. That's why not all of the microorganisms present in the environment will grow in the lab. Okay, so this is basically a summary for this particular objective. So out of the 385 distinct isolates that we tried to purify, okay, only 235 isolates were successfully purified. Most of the isolates became viable but non-culturable. So we assume that there are certain conditions in the culture media, certain cofactors that are not being met. That's why they failed to grow. Okay, notice on the bottom row that the number of colonies per sampling site is not statistically different. Now, this could be due to the fact that the microclimate within these sampling sites are not so different. Okay, for objective 2, which is to isolate or to screen the isolates for cellulase activity, we did 2 assays, the carboxyl methyl cellulose agar assay and the filter paper activity assay. So for the CMC agar, so basically what we did here was we cultured all of the isolates in agar plates, and then after 1 week, we made agar plugs, and then we imprinted that on the surface of CMC agar. And then after incubation for 48 hours, we flooded the plates with iodine, as you can see in the figure, and then measure the zones of clearing, which is an indication that the CMC is actually being degraded. Okay. And then for the next assay, for the filter paper assay, the first thing we did was to prepare the CFS, or the cell-free supernatant. So based on the result of the CMC agar assay, we chose isolates which had very large zones of clearing, about at least 20 millimeters of zones of clearing. And then we processed, or we inoculated them through to a medium called YMB, and then we subjected that to submerged state. We inoculated them to CMC broth, no, not YMB. That's for a different study. So we inoculated the isolate to CMC broth, supplemented with 0.2% glucose, and then incubated that for about a week. Okay, and then afterwards, to do the actual assay, we mixed a zero point 0.5 ml of CFS with 50 milligrams of filter paper in sodium citrate buffer. And then we terminated the reaction by adding DNS, and then boiling the samples, diluting it with water, and then measure the absorbance at 540 nanometers. Now, for the calibration curve, we did the experiment with diluted concentrations, a serial dilution of glucose as standard. And then we used this formula to calculate for the FPA's activity. So this formula simply gives you the amount of substrate that is converted to glucose by cellulase that is present in the CFS over the course of 60 minutes. Okay, now for the results, so these are just representative figures of our CMC agar. And based on this particular assay, only 114 isolates demonstrated a zone of clearing with this assay. Now, um, we were able to isolate bacteria that secretes cellulase, because soil bacteria are known to decompose organic matter, and cellulose is the most abundant organic matter possibly present in the soil. Okay. Now, as for the filter paper activity assay, we only found two isolates. Okay, with such activity, you have EWLG2 and EPNA9 with F-pace activity of zero point 0.6 and zero point 0.9 respectively. Now, notice that many of the isolates had no detectable activity for the F-pace assay. Now, this could be due to a low concentration of cellulose, because our conditions were not optimal for their production. And it could also be due to the type of cellulase produced, because some cellulase are surface associated, meaning there's a possibility that they are not actively being secreted into the culture media. That's why the cellulase concentration in the CFS could be low. Now, fortunately, the FPU we obtained were comparable to a previous study by Liang in 2014, and were actually higher compared to the results presented by Gupta in 2011. Now, unfortunately, the FPU we reported is much lower than those reported by La in 2012. Now, this could be due to a difference in the substrate used. In this particular experiment, we used filter paper, but in their experiment, I think they used corn kernels. And there's also a difference in methodology, because in the work of La et al., they had to concentrate the CFS. And in doing so, you would have a higher concentration of cellulase, which would translate to a higher FPU activity. Okay, now for the third objective, which is to screen the isolates for antimicrobial activity, we did three assays. We have the agar plug, the agar well, and the broth micro dilution assay. Now, for those not familiar with the agar plug diffusion assay, this is basically where you grow your isolate, and then after you have a sufficient growth, you make a plug by boring a cork borer through the agar and then transferring that agar plug into an MHA plate seeded with different test microorganisms. And for this experiment, these are the different test microorganisms that we used. We have representatives of gram-positive bacteria. We also have some gram-positive, gram-negative, and we also have some fungi. Okay. Now, isolates that had significant activity in the agar plug assay, meaning if their zone of inhibition was more than 20 millimeters, we selected them to proceed to the next assay, which is the agar well. So for the agar well method, basically what we did here is that we inoculated these isolates into YMB broth and then incubated that. After incubation, we centrifuged to obtain the CFS, and 50 microliters of that CFS was placed in wells, okay, in wells as seen in the image, but the plates were previously lawned as well with the different test microorganisms. And then for the last experiment, we did the broth microdilution assay. Now, the first part of this experiment, this is actually a long assay. Okay, matagal na akong ginawa ni Joma. But the first thing he did was to optimize the CFS. And to do that, he experimented with three different media. We have M9 or minimal salt medium, and then M9 plus 2% glucose, and YMB medium as well. And then and then afterwards, so what he did was he inoculated these different media with different isolates that showed good activity, and then they incubated that for 14 days. For 14 days, and then for each day, what he did was he prepared CSF and then tested them using the agar well assay. Okay. So based on that optimization effort, he was able to determine that the best medium for this particular assay would be the YMB medium. So in preparing the crude extract, he seeded 2.5 liters of YMB and then incubated that for 6 days, because based on his optimization experiment, which lasted for 14 days, the activity started to decline after the sixth day. And then after 6 days of incubation, he prepared the CFS and then mixed that with ethyl acetate, and then he collected the solvent phase and then reduced that. He dried, okay, the paste, and then dissolved that in 100% DMSO to make a working to make a stock solution of 30 milligrams per ml. Now, after he had the crude extract, he can now proceed he then proceeded with the broth microdilution assay. So this is basically where you expose a standardized amount of microorganisms to a serial dilution of the extract dissolved in DMSO. I think the range of the serial dilution was from 500 micrograms per ml to 1 micrograms per ml. And then he incubated that and then determined the MIC. MIC stands for minimum inhibitory concentration, or the concentration or the lowest concentration where you no longer have growth of the test microorganism. Okay. Okay, now for the results and discussion, these are just some representative plates of the agar plug diffusion assay, and these are the results. Now, to summarize the results, only 18 out of 52 isolates had zones of inhibition, and out of these 18, only 10 had zones of inhibition against bacterial test pathogens, which ranged from 12 to 23 millimeters. And as for the fungal test pathogen, only 14 isolates had zones of inhibition, which ranged from 8 to 21 millimeters. Now, why do microorganisms in nature produce antimicrobial? This is basically to give them an advantage against their competitors. But based on our study, a lot of the isolates exhibited no antimicrobial activity. Now, this could be due to the following. Maybe they don't really secrete, or they don't really produce um, antimicrobial, or it could be due to that fact that the amount of my antimicrobial produced was low because we did not provide the best conditions for those particular isolates, or because the antimicrobial had a very low rate of diffusion through the agar. Okay. Now, for the agar well diffusion assay, these are just some representative figures. So you can see the zones of inhibitions. Some isolates also did not have any activity. Now, for the summary of the results, only seven isolates had activity uh, against the bacterial and the fungal isolates. Two isolates had activity against bacteria, and then you have five isolates which had activity against the fungi. Now, the CFS for this particular experiment was collected on or after the seventh day of incubation. We did the same experiment but collected the CFS after 14 days, but unfortunately, we lost the activity against the bacteria. Now, some activity for the fungi remained, but as you can see, if you compare the two figures, okay, the activity against the fungi was significantly lower after 14 days. Now, several isolates had no zones of inhibition. Now, this could be due to the media that we used. Maybe it was not optimum for the secretion of their secondary metabolites, or it could be due to the amount of metabolites being secreted, which is low. Now, regarding the loss of activity after 14 days, this could be due to the degradation of the antimicrobial itself. Okay, now for the micro broth dilution assay, this figure just shows you a part of the optimization experiment performed by JOMA, showing that the CFS made with YNB inhibited the growth of Candida albicans and Candida tropicalis. Notice also, based on the graph, that starting on the sixth day or sixth or seventh day, the activity already started to decrease, which is the reason why we stopped the incubation for generating the CFS for the crude extract on the sixth day. Okay, now this is the result of the MIC. So as you can see, you have I think one, two, three, four, five, uh, six isolates had activity against fungi, and then two isolates. Okay, three isolates had activity against bacteria. Okay. Now, for the discussion, notice that the MIC we got for the isolate was significantly higher compared to the controls. For the isolate, our MIC values were 31 micrograms per ml to as high as 500 micrograms per ml. Now, you compare that, let's say, with penicillin or nystatin. Now, this is understandable because we were working with crude extracts and not pure compounds. Okay. Now, notice also that we observe more antifungal activity than antibacterial activity. Now, this could be a reflection of the microbial interaction occurring where these isolates were obtained. It's possible now within those areas, fungi are a more serious competitor than bacteria. Another reason could be due to the conditions we used for the antimicrobial production, which may not be optimum for these isolates. Now, for the last objective, okay, which is to characterize and identify promising isolates, we use two general methods: morphological and biochemical characterization and molecular identification. For the molecular and biochemical characterization, we did the following experiments. The first two are actually the most important, the gram stain and the catalase test, because we had to make sure that all of the isolates we used for the previous assays were all gram positive and were catalase positive as well, because actinomycetes, bacillus, and streptomyces are all gram positive and catalase positive. The other test listed here is basically just to help us differentiate the isolates from one another after we identify them using the molecular identification. Now, for the molecular identification, these are the basic steps. We did DNA extraction using the boiling methods of Valley et al. In 2012. And then we did a PCR using universal primers 27F and 1492R. And these are the PCR conditions we used after optimization in the lab. And then the PCR products were sent to Macrogen for DNA sequencing, after which we did the sequence analysis. We used BLAST-N in NCBI to identify the isolates as either Bacillus or Streptomyces. And then afterwards, we downloaded 16 srRNA sequences. This is for the generation of the phylogenetic tree. So we downloaded 23 sequence types for Bacillus and 48 sequence types for Streptomyces. We used a bioedit to align the sequences and to trim the sequences. And then we used the J model and the Zia test to evaluate the nucleotide substitution and the nucleotide substitution saturation, which would eventually affect our interpretation of the trees. Now, the phylogenetic trees were generated using pulp, and the trees were rendered using Tree Explorer. Okay. Now, for the results, these are the characteristics of our selected isolates. Notice that we limited the characterization and identification to those that we felt exhibited high cellulase activity and high antimicrobial activity. So as you can see, all of the isolates listed here are all gram positive. Okay. Okay. And then most of them are actually catalase positive as well, with the exception of this. So I made some basic searches, and I found out that not all streptomyces are actually catalase positive. There are certain species of streptomyces that are catalase negative, which is consistent with the results that we got. Now, for the identification of the selected isolates, so we were able to identify those that exhibited cellulase activity as Bacillus cereus and Bacillus pseudomycodes. Okay, so the percent identity is well beyond acceptable. Now, for the antimicrobial activity, we were able to identify one, two, three, four, five, six isolates belonging to the genus Streptomyces. We have there Streptomyces olivaceus and Streptomyces exfoliatus, and then one isolate, EPLG5, as Bacillus cereus. So again, the percent identity were beyond the acceptable value, and we have 100% query cover. Now, for the short discussion, okay, so we were not really surprised that we were able to identify a species of Bacillus for the cellulase activity, because this particular genus produces about 60% of all available enzymes, not just cellulase. However, we have to point out that this is the first report of cellulase activity for Bacillus pseudomycodes. Now, the same thing is true for Streptomyces, because this particular genus is said to produce about 67% of all clinically useful antibiotics. Now, please take note that based on our sequence analysis, we have here 4 isolates, okay, that were not identified to the species level. So they might represent novel species, but I have to be careful with that statement, because in the phylogenetic tree that we made, we only used a 48 sequence types, when in fact there are over 550 species of Streptomyces currently named. Now, these are just phylogenetic trees that we generated to confirm the identification of our cellulase producing isolates. So the same thing is true for our antimicrobial producing isolates. Notice that the bootstrap scores are quite high, especially for those isolates that were identified to the species level. So in conclusion, okay, so we concluded from this study that green spaces are actually rich sources of bacterial isolates with cellulase and antimicrobial activities. Now, these isolates can be studied further in the hope of helping address problems with fuel shortage and with antimicrobial resistance. Of course, this is just basically a very basic step towards that direction, and many more experiment needs to be done for these isolates to be of industrial and actual medical use. So for the acknowledgement, I would like to acknowledge the NSRI and BOST PSHERD for funding this project under project number PSHERD Bio 1704, and of course the Institute of Biology. I have to say also that Mr. Dakiwag already sent the first part of this work, the cellulase producing part, to the International Journal of Microbiology, and we recently received the review of the article and they are requesting revisions. So hopefully after we revise the manuscript, they would consider our manuscript for publication. So thank you very much, and I am looking for your questions during the open forum. Thank you.
Thank you so much, Sir Hill. Can we have a round of applause, a virtual round of applause for Dr. Penular? Thank you so much, sir. So we have just had two very interesting talks, a bit heavy on the technical side for some of us. There are many from the high schools and colleges that may have questions for our two speakers. Actually, there are a number here. And shall we start? Dr. Penular and Dr. Ubusan?
Hi, ma'am. Yes.
Hi. Yeah, so thank you so much once again for these. We have the first question. It's for Dr. Obusan. We'll dive right in. So the first question is, what are the biotechnological applications of the enzymes that have those that you mentioned, like the tyrosine, the lactase, and I guess the next question is related. Why is there a need to monitor enzymatic activity in momors?
Okay, so I will answer first the last question. As I have explained, Momors is a stressed environment because of the presence of many pollutants. So an example of pollutants are phenols and indole substances, for instance, which are actually converted into, for example, melanin through oxidation. Specifically oxidative polymerization. So we isolated bacterium that has the ability to produce the melanin pigment, and we infer that that ability is part of its adaptation to that particular environment. So there is a melanin production because there is the presence of tyrosinase enzyme that catalyzes the reaction of those phenolic substances, which enables melanogenesis of the bacterium. So I would like to uh, deal with the tyrosinase as an example. Some of the biotechnological applications, it's used as biosensor, also in metal chelation, and also the role of tyrosinase in providing a neuroprotective effect on patients with Alzheimer's and Parkinson's disease is being explored right now. As for lactase, we did not detect lactase activity, but in general, lactases are important in the industry of dye degradation and degradation of paper and other similar products, and also in the production of pigments that can be used in the textile industry.
Very interesting. Thank you so much. To whoever posed those questions, if you have succeeding questions or for anyone who has a question, huwag kayong mahiya. Go ahead and type your questions in sa Zoom chat box or in the Google form for the YouTube watchers. Our next question is for Sir Hill. And the question is is there a biotech application for cellulase production or of cellulase production?
Oh, correct. So the main biotech application for cellulase degrading bacteria would be in the production of biofuel. Of course, there are two types of biofuel. You have bioethanol and biodiesel. Now, microorganisms that produces cellulase are useful in this aspect because they can serve as biocatalysts or bioprocessors of biomass containing cellulose, which can be converted to glucose, which in turn can be used to produce these biofuels. So that would be the main application of these
Of
microorganisms.
them, yeah. Okay. There's another question for you, sir. Um, do you think that the widespread use of antimicrobials contained in personal care products contribute to antimicrobial resistance?
Oh, definitely. Okay, so a lot of the things we commonly use, so let's say from liquid detergents to hand sanitizers, some of these contain antimicrobial agents, which technically are their concentrations are sub they're minimal, meaning they are not optimum or to actually to kill microorganisms. And when microorganisms are exposed to these concentrations, to these suboptimal concentrations of antimicrobial, the tendency for them is to actually develop resistance over time. So again, the main reason why antimicrobial resistance has become a problem recently is because not just of antibiotic overuse, let's say in the poultry industry, but also antibiotic misuse, not just in the clinical setting, but also in our day-to-day activities. So again, we have to be careful when using hand sanitizers, or for example, when we're under medication, make sure that you complete the medication so as not to contribute to this serious problem.
Hmm. So this is very interesting. I mean, I don't know about the other participants, but not finishing the antibiotic course that was prescribed to you has always been, the doctors even say that it might lead to resistance, and then the antibiotic won't work anymore if you use it again. But this problem with hand sanitizers and other personal care products, will this, do you think, will this become a big issue in the near future? Everybody is using ethanol now and hand sanitizers everywhere, spraying everything because of COVID. Um,
Maybe
do,
not, okay, okay.
yeah.
Maybe not for ethanol, but for distinct compounds or agents within certain materials, let's say like triclosan, for example, and other substances. So, of course, when we ultimately the bacteria that is basically washed off after using these personal products, they go into the environment. Okay, and we get sick because we become infected by microorganisms from the environment. So it's actually a cycle. Everything is connected. So not just because we wash off these microorganisms or disinfect, but if some of them survive, okay. Diba remember a lot of advertisements claim
Right.
99.9% efficiency, effectiveness, 99.9. But what happens to that 0.1%? That 0.1% after 24 hours grow or multiply into millions and billions of cells. So again, of course, don't have to criticize yung claim na 99.9%. But again, leaving that 0.1%, that 0.1% might already be the one contributing to antimicrobial resistance.
Okay. So, well, 99.9 is better than a less percent, I
Of
suppose.
course, absolutely,
Yes.
absolutely.
Okay, thank you so much, Sir Hill. This next question is for Dr. Christine. What method would you recommend to create a water with heavy metal content for an assay to assess growth of fungi?
Yes, actually, I sent also my reply
Oh, okay.
back. So I think Carlo is asking a method to test the effect particular heavy metal on microorganisms. So he mentioned fungi, for instance. So usually the heavy metal is incorporated in the media, but the methodology must be carefully designed and approved by the Biosafety Committee and Pollution Control Committee of the institution. It depends also on your target heavy metal.
I
And of
see.
course, there are parameters of growth that you can test, and you can also check for tolerance mechanisms.
Mhm. Okay, Carlo, if if you have further questions for Dr. Ubusan, feel free to ask some more. Um, Huwag kayong mahiya magtanong. Nandito sila. Let's take advantage of their being here. Ako, I have a question. I'm sorry. I hope this doesn't put you on the spot. For both Sir Hill and for Ma'am Christine, we have a lot of students in our audience, and many of them may be trying to think of science projects or research topics. Are your labs open to helping out students who would like to work on the topics that you do research on? Who would like to go first?
Ladies first.
Okay. Well, that's a good question actually, kasi a lot of students are sending emails talaga or asking us if we can help with the identification of their bacteria or any assay. So as of now, kasi especially in the case of the Institute of Biology, we have, and UP in general, we have restricted lab access. And so we're also finding it difficult to deliver the outputs of our research projects. We can assist students online, like they can ask questions in relation to the theoretical background of the study that they're working on kung kayang gawin, maisingit ng researchers or research associates. But we have very limited time right now because of the community quarantine. But of course, we can give advice. You can email us. The contact details of the faculty of the Institute of Biology are in the website. And you can also ask for some papers. Sumasagot naman kami sa email. So, mas matutulungan, in my case, mas makakatulong ako ngayon sa online discussion, sa
Okay.
virtual intervention and extension work.
Thank you so much, Ma'am Christine. Sir Hill?
For my part, similar to Dr. Ubusan, the mandate of the Institute of Biology is not just teaching research, but also extension. Under normal conditions, a lot of the faculties in the Institute of Biology are willing to accept services from students that are not from UP. I have, for example, in the past, I have helped a lot of students conduct their experiment, but unfortunately, because of the situation we have right now, a lot of things are restricted within the laboratories. No? So, for example, we limit the number of individuals that can go to the lab. As a matter of fact, all activities related to undergraduate thesis and graduate thesis is actually not allowed in the Institute of Biology. So for those students who send me emails asking for help, I normally give them the same explanation, but at the same time, I usually refer them to NSRI. So I'm not so sure the situation in NSRI, but I think at least two labs in NSRI before should be able to help them. I usually refer them to BRSL and to MRSL. So I'm not so sure if those two labs are actually willing to accept services at this time. So thank
Okay, so to the students out there, our speakers from the Institute of Biology are open and willing to help in a limited capacity because of the quarantine. So they have let you know how you can contact them. Also, as Sir Hill mentioned, perhaps some labs here in NSRI can help you out. So you also have our contact details on the web. We have time for just one last question. This is for Mom Christine. And it says, I have a fungal isolate that produces an unknown pigment. I am currently proposing a research for it. I am thinking of applying de novo assembly of the genome and functional annotation as well as identification of the pigment. Would it be better if I look into the genes first prior to identification of the pigment or the other way around?
Okay. I would advise to first elucidate the nature of the pigment produced by your isolate. Actually, in our case, we went through uh, several iterations. So at first, we thought that the pigment that we are producing is in relation to lactase activity. And so we kept on repeating the experiments and we're not able to replicate the lactase pigment production result that we had until we discovered that it is a different type of pigment. So what we did is that we extracted and purified the pigment to know if it's really, worthy or significant for us uh, to 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 focus our efforts on that isolate. So we isolated the pigment and then we submitted it for chemical characterization. No, we did the chemical characterization, but we submitted our sample for FTIR analysis for us to know the absorbance spectrum and to relate the significance of the pigment that we have in relation to the other types of pigments with known industrial applications. And also you need to work first on the yield. Okay, so you need to know if you are producing a significant enzymatic activity. You need to know first what is the enzyme that catalyzes the production of the pigment, and then you need to know if the pigment production is significant enough. And the whole genome sequencing is actually quite expensive. In our case, the reason why I acknowledge the Philippine Genome Center is that they provided the whole genome sequencing for free. So
Wow.
abangan nyo rin yun mga students kasi 2 years ago, they announced their call for the submission of isolates that you think can be considered for free sequencing. So you need to submit the abstract and some details of your study, why you think your isolate must be given the free treatment for sequencing work. So yun yung nangyari doon sa amin. And then after that, we performed the usual whole genome analysis. So what I'm trying to say is ang mahal-mahal kasi ng whole genome sequencing. So may mga paraan pa naman, especially kung wala ka pang research budget as of now, may ibang mga paraan pa naman for you to really evaluate the pigment production and other activities of your ICD.
Oh. Thank you so much, Ma'am Christine. I'm sure the person who asked that question learned a lot. Napaka-intricate pala, ang daming steps na kailangan i-consider. So thank you so much. And that brings our open forum to an end. Anyone who has a question for Dr. Christine or Sir Hill, both of our speakers said that you can contact them via email. So feel free to use that to use that means of communication. So the NSRI would like to thank our speakers in a special way. So may I call on Sir Tux, Dr. Estacio, to present the certificates of appreciation.
Yes, ma'am. I'm here. May I request ma'am to please flash the
Yes,
certificate?
Sir Jude.
All right. So, hello. Okay, so this certificate of appreciation is hereby granted to Dr. Hill M. Peñular for delivering the talk entitled Cellulase and Antimicrobial Activities of Bacillus spp and Streptomyces spp isolated from the urban green space in the Philippines during the NSRI Lecture Series 2021 held on the 29th of January 2021, 2 p.m. to 4 p.m. via Zoom. Signed, Elmer Estacio, NSRI Director. Thank you very much, Hill.
Thank you, sir.
Thank you. Thank you, Hill. Nice to see you again, Hill. Long time no see.
Oo nga po, sir. Thank you.
Shall we have a show of hands for Sir Hill? Okay.
And secondly, this certificate of appreciation is hereby granted to Dr. Marie Christine M. Obusan for delivering the talk entitled Degradation, Potential, and Enzymatic Activity of Bacteria from the Highly Polluted MMORS during the NSRI Lecture Series 2021 held on the 29th of January 2021, 2 to 4 p.m. via Zoom, signed Elmer S. Estacio. Ma'am, thank you very much po. Such a nice talk.
Sir.
Thank you.
Yes. Let's clap our hands virtually for Dr. Obusan. Thank you so much. Sir
Thank you
Tux,
so
thank
much.
you.
Ma'am, thank you din po. Congratulations, NSRI team.
Okay, I would like...
Maraming salamat.
Yes, sir. Thank you then, sir. I would like to remind everyone that there will be a photo documentation session after the program, and that there is a link to the evaluation form. It will be posted on the Zoom chat box and in the YouTube description box. Everybody, please make sure to fill up the evaluation form within the day so that you can receive your e-certificate. Now, for the last part of our program, I would like to introduce to you this year's chair of the NSRI Seminar Committee. She is a university researcher too and a certified specialist microbiologist. As Deputy Technical Manager of the Microbiological Research and Services Laboratory, or MRSL, of NSRI, she has been instrumental in developing and strengthening the technical capability and capacity of the MRSL, as well as other government agencies and private companies as a member in technical working groups and as a trainer. To formally close this afternoon's webinar, let's all please welcome Ms. Vina B. Argayosa. Ate Vina? Nakamute pa po kayo, Ate Vina.
So good afternoon, everybody. We are ending this webinar with the promise that we will have more this year, since the NSRI Seminar Committee plans to do this quarterly, as was already mentioned by our director. So as we continue to do these webinars, we are opening and enriching the minds of budding young scientists like you, some of those who are listening, and adding to the ideas of mentors, as well as giving a platform for exchanging ideas and experiments. So in other words, we are contributing to build a culture of science in our country. Talks like this have been part of the NSR experience through the years, but starting October last year, 2020, this format allowed for more access and more people can hear and listen to these projects. We would like to thank our scientist speakers. Our directors spent time with us and all of you who watched this seminar with us. Behind the scenes, I would like to thank every member of the NSRI Seminar Committee who worked the tasks and learned along the process. Each member comes from different laboratories, as you can see with the logo below, and offices of the Institute also. And each one has a valuable contribution. Thank you very much, team. So it is now 2021. We are here from 2020, and we look for hope as we continue to trust God with our lives, right? And to perform our safety protocols. Don't forget that. Good afternoon to all. And remember to click the link to the evaluation form so that you will receive your certificate of attendance. Okay, so now we will prepare for our photo documentation. So here is the evaluation form with the link, but you will also find one in your chat box. And for those for the YouTube participants, I think it will also be in the description box for you to access this evaluation form link. So we will now call on Kuya Choi for our prepare everyone for our photo documentation. If you may want to turn off your videos.
It's so nice to see that there were more than 100 participants at the height of the webinar. Yes.
So is this the okay? So Kuya Choi, can you please take a photo?
Yes, ma'am. Second part na po.
So, kamusta na Kuya Choi?
Last picture.
Last.
Okay na po, ma'am.
Okay, salamat sa lahat sa pagdalo ngayong webinar. So, expect the next webinars. The announcement will be given sa NSRI Facebook page.
Thank you.
Thank you, everyone.
Thank you very much.
Thank you.
Congratulations.
Thank you.
For those of you who had difficulty with the evaluation form link a while ago, try it again. It was disabled a while ago, but it's working now. So give it another try. Thank you. See you all next webinar.
Thank you po sa lahat. A good day and a happy weekend to all.